Okay, just copied on this question. Am I audible? Hello? Hello?
Sure.
Okay, just copied on this question. So this is chapter 2. Okay, in module 1 this is chapter 2. Okay, just write on the question. Draw a neat cross-sectional view. Draw a neat cross-sectional view of eye human eye and label its parts. And label its parts. Or with a neat diagram. With a neat diagram of the eye, I explain the human visual system working. I explain the human visual system working. So if we consider the cross-sectional view of an human eye, it would look like this. Okay, so this portion, okay, this portion will be called as anterior portion, and this portion will be called as posterior portion. Okay, so this is your anterior portion. Okay, this is your anterior portion, and here this will be called as posterior portion. Okay, so this will be called as your posterior portion. Okay, so the eye, okay, this is around 20 millimeter in diameter. So this is somewhat spherical in nature. Okay, so there will be three membranes. Okay, there will be three membranes which will be covering the eye. So the outermost membrane is the cornea. So the outermost membrane is the cornea. The same membrane, okay, here it will be called as cornea in anterior portion, and the same membrane at the posterior portion it will be called as sclera. Okay, so the outer membrane itself, okay, it will be called as cornea in the anterior portion, and it will be called as the sclera in the posterior portion. Okay, the cornea is very tough and it is a transparent tissue, whereas the sclera is opaque. Okay, it is an opaque membrane. Okay, next to this membrane, okay, so there is a membrane called as choroid. Okay, so this is the second membrane which is present inner to this sclera membrane. So this is your membrane which consists of fibrous networks which will be helpful to supply blood to the uh, eye. Okay, so it consists of blood cells that serve as a major purpose for the source of nutrition to the eye. Okay, so here it is called as choroid. The same thing, okay, the same choroid. Okay, so the same choroid which extends in the anterior portion. Okay, so it will be forming the ciliary body. So it will be forming the ciliary body and the iris. Okay, so the choroid membrane itself, okay, in the posterior it's called as what, choroid. When the same thing goes to the anterior portion, it forms the ciliary body and the iris. Okay, so the ciliary body will be used to hold the lenses. Okay, and the iris will be contracting or expanding to control the amount of light which will be entering into the eye. Okay, the central portion, okay, the central portion of the eye is called the pupil, which varies in diameter from about approximately 2 to 8 millimeter. Okay, so this lens, okay, whatever the lens which you are seeing over here is made up of concentric layers of fibrous cells, and it is suspended by the fibers, okay, that attach to the ciliary body. Okay, so the main function of the ciliary body is to suspend this lens by holding on to this. Okay, so this lens, okay, this lens contains about 60 to 70% of water, okay, and 6% of fat. Okay, so this is the uh, one which will be containing more amount of protein than any other tissue in the eye. So this will be containing the major amount of protein. Okay, the innermost membrane. Okay, so now we have come to know regarding cornea. The same thing here, it will be called as sclera. The next membrane is called as choroid. Okay, inside the choroid, okay, there is a membrane called as retina. Okay, there is a membrane called as retina. So this is the innermost membrane, which is the retina. Okay, so when the eye is properly focused, okay, whenever you need to see an object, okay, let us consider there is an object over here. Okay, if there is an object over here, okay, let us consider there is a tree. If you need to see this object, okay, the light from this, that is the reflected light from this, has to fall on the eye. Okay, the light enters through this lens, and it has to be focused. Okay, it has to be focused on the retina. Okay, it has to be focused on the retina, so that the image is formed within your eyes. Okay, so when the eye is properly focused, light from an object outside the eye is imaged on the retina. Okay, so able to visualize the pattern of the image. Okay, when you see at an object, you'll be able to find out the pattern. Okay, if you see at this object, you'll be able to detect the tree. Okay, there should be some pattern for every object. Okay, so the pattern vision, okay, is affordable just because of the light receptors. So there'll be receptors. Okay, there'll be receptors. Okay, so there will be receptors which will be, so there will be which will be present over this retina, which are responsible for the pattern vision. Okay, when I come for receptors, so there will be two types of receptors. One is called as cones, okay, and the other is called as rods. Okay, so these are the cones, and the second one is called as rods. Okay, so these are the two receptors which are which made us to visualize the pattern. Okay, so this out of these two, the first one is called as cones. So one which enables us to see the colors of the object, so it's also called as photopic. So this will be called as photopic. Okay, so in the presence of bright light, okay, you'll be able to see the color of an object. It is just because of these receptors, that is cones. Okay, similarly in the dim light, okay, in the dim light you'll be able to see the object, but the pattern of the object or the color of the object will not be visible clearly. Okay, so this rods is also called as scotopic. It will be called as scotopic. Okay, so there will be two types, one is photopic and the other is scotopic. So the cones are the photopic and rods are the scotopic. So this will be present on the innermost layer, okay, retina. Okay, so usually the cones, okay, when we come here, the cones will be present near the fovea. Okay, so it will be present near the fovea. Okay, so the fovea is nothing but the exact center of the eye. Okay, the cone will be present near the fovea, and at the outermost boundary, okay, at the outermost boundary will be having the rods. Okay, it will be having the rods. Okay, you just need to remember that cones is responsible for photopic vision and the rods is responsible for scotopic vision. Okay, photopic means in the presence of brightness, scotopic means in the darkness. Okay, next if we come over here, this is the center which is called as fovea. From here to here, okay, there will be receptors available. Again, there is a gap over here. Okay, if you clearly observe here, there is a gap over here where the nerves enter into your eyes, okay, to provide nutrition to the eye. Okay, so there is no retina membrane from this location to the end. Okay, so here the nerve enters into the eye. Okay, so since there are no receptors, okay, whenever the reflected light falls on this, okay, this area, no receptors, will not be able to identify that object. Okay, so that will be called as blind spot. Okay, it will be called as blind spot. So whenever the reflected light falls on this, you will not be able to see that particular image, so it's called as a blind spot. Okay, clear with this? Hello, hello, hello.
Yes, sir.
Okay, so this was regarding the cross sectional view of the human eye. Okay, so as I said, so this is the central fovea, so let us consider this as 0 degree. Okay, so let us consider the distribution of rods and cones. Okay, distribution of rods and cones in the retina. Okay, this is the 0 degree. So as I said, rods would be concentrated heavily at the fovea. Okay, so here the continuous line. Okay, so the continuous line indicates what, cones, and the dashed line, okay, or the uh, so this is continuous line, uncontinuous line or dashed line indicates the rods. Okay, so cones, so at 0 degree, if you observe here at 0 degree, so there is a major concentration of cones. Okay, it decreases gradually when you go away from the center. Okay, so it will be having very high concentration, it will be having very high concentration at the center, but it goes on decreasing as it goes away. Okay, similarly if you consider the rods, okay, it will be having higher away from the fovea. Okay, this is the concentration of which one, this is the concentration of rods, this is the concentration of rods. Okay, if we consider here from 0 to 20 degree, there will be concentration of what, there will be concentration of rods from here to here. Okay, if you observe here, there is no rods, there is no cones. Okay, the reason is there is no receptors, means it's a blind spot. Okay, so we'll be calling this as blind spot just because there will be no rods. Okay, so just because the nerve enters into the eye at this location, so it doesn't contain any receptors. Okay, so this is the degree starting from the fovea, so, and the distribution of cones and rods. Okay, clear with this? Hello, hello.
Yes, sir.
Okay,
Sure.
next question. Briefly explain the principle of briefly explain the principle of image formation image formation in the human eye. Briefly explain the formation image formation in the human eye. So now let us consider an image, okay, of a tree which is around 15 meters high. Okay, when you look at this object, okay, the inverted version of this image, okay, the inverted version of this image is available on the, okay, on the retina surface. Okay, it will be available on the retina surface. So when you see at this object, okay, when you see at this object, this is at a distance of 100 meters. Okay, it is at a distance of 100 meters from the focal point. Okay, it is at a distance of 100 meters from the focal point. Okay, so the inverted image, okay, the inverted image will be formed in the eye. Okay, this inverted image will be passed to your brain. Okay, then at the brain it again inverts image, and it again inverts the image, and you will be able to see the proper image. Okay, if you are asked to find the height of this inverted image, okay, if you are asked to find the height of this image, this is the formula which you need to make use of. Okay, so the height of this is how much, 15 meters. Okay, the height of the original image is what, 15 meters. It is at a distance of 100 meters. Okay, it is at a distance of 100 meters from the focal point, which is equal to, so the height of this, okay, let's consider the height of this as h. Okay, so h divided by 17 meters. Okay, so this is the distance. okay. This is the distance which is 17 meters, okay. Here you need to compare the camera, okay. You can compare a camera with a human eye, okay. So in case of camera, what we will do is the focal length, okay. The focal length would be fixed, okay. So sorry, the focal yes, the focal length would be fixed, and the imaging plane can be varied, okay. So the focal length will be fixed and the distance between the imaging plane and the lens can be varied. You might have observed, okay, they will be moving the camera forward and backward. The lens would be moved forward and backward to take the image, okay. If the lens is at a far distance, they will be moving the lens forward, okay. So they will be adjusting the lens so that it can be filmed on the imaging plane. Here you will not be able to move the eye forward and backward, okay. It is fixed, okay. So what you can do is you can vary the focal length itself, okay. You can vary the focal length. By doing that, be able to take the image, okay. So the difference between camera and the human eye, okay. In case of camera, this distance, okay, this distance can be varied, okay. Whereas in case of human eye, this cannot be varied, okay. It is fixed to 17 mm, okay. So h divided by 17 mm, okay. This is your h, the height which you need to calculate, divided by 17 mm. So this is the fixed distance, okay. So that gives you the value of h. So that is 2.55 mm. So the height of the inverted image which is formed on the retina will be around 2.5 mm, okay. So this is how the image would be formed in the eye, okay. So clear with this?
Yes, sir. Yes, sir.
Okay, the main thing is what? In case of camera, the distance between the imaging plane and the lens can be varied, whereas focal length will be fixed, okay. In case of human eye, focal length can be varied, but the distance is fixed, okay. Next, write down, explain the importance of brightness adaptation and discrimination in image processing. Plot the typical Weber ratio curves. Typical Weber ratio curves. So this is very important question from the point of exam, Weber ratio curves. So here, as I said earlier, so there will be two types of vision. One is called as a scotopic and the other is called as photopic, okay. Photopic means in the presence of bright light, scotopic means in presence of darkness, okay. So you might have observed that when you enter from a bright room into the dark room, you will not be able to see the objects properly, okay. It takes some amount of time to adjust to the darkness, okay. So that is nothing but adjusting to the darkness from brightness is nothing but we will be calling it as brightness adaptation, okay. You need to adjust from brightness to darkness. The amount of time it takes to adjust your eyes from darkness or brightness to darkness is called as adaptation, okay. So in this image they are taken an example of adaptation from darkness to brightness, okay. Or you also can also take from brightness to darkness or darkness to brightness. For example, if you enter into a film theater, okay, after 10 minutes, okay, You'll be entering from brighter region into darker region. Okay, the moment you enter into the film, you'll not be able to see the seats, right? Hello, hello, am I audible? Hello.
Yes, sir.
Yes, sir.
So
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
when you enter into a film theater, okay, after 10 minutes, you'll not be able to see the seats properly, okay, just because you entered into dark room from the brighter region. Okay, after several seconds, okay, you'll be able to see the seats. Okay, this is just because your eyes needs to adapt from brighter region to darker region. Okay, so this is called as adaptation, brightness adaptation. Okay, the same thing is shown in the figure over here. Okay, so this is the intensity. Let's consider this is the zero intensity level, and as you go towards left it is darkness, when you go towards right it is brightness. Okay, as you know that in brightness it will be operating, photopic vision will be operating. So here which sensors will be operating? Here cones will be operating. Okay, here cones will be operating, whereas in case of darkness, towards left it's darkness, scotopic will be acting, so the rods will be responsible for scotopic vision. Okay, and so here if we consider here this is your scotopic vision, and your photopic vision is this line. Okay, this line, the intersection of these two. Okay, the intersection of these two indicates the brightness adoption. You are adopting from dark to bright. Okay, this is the location where your adaptation takes place. Okay, they are given one rough curve. Okay, let us consider you are entering from this location. Again, this is the dark and you need to go into the bright. Okay, this is the curve which you are given. So this is the adaptation which takes place from dark to bright. Okay, so when the light increases, okay, when the light goes on increasing, you'll not be able to see the brightest light. Okay, can you see into sun? It is impossible, right? Just because it is very much. So that limit in the brightness is called as a glare limit. Okay, there will be a limit to see in the brightness also. The more higher threshold is called as glare limit. Okay, again in the darkness there will be some limit. Okay, beyond this level will not be able to see even in the darkness. Okay, so this is the brightness adaptation which will be done by the human eye. Okay, clear with this? Hello, hello, clear with this?
Yes, sir.
Okay, so the same thing. Okay, will be using Weber ratio. Will be using Weber ratio to indicate the discrimination between the brightness and the darkness. Okay, let us consider an experiment to do this. Let us take a rectangular or square surface and illuminate this complete surface with light I. Okay, the intensity of the light will be I. Okay, for a moment you observe there is no circle over here. Okay, circle over here. Okay, you will be illuminating this square with a light with an intensity of I. Okay, so the entire surface will be illuminated with an intensity of I. Okay, after that you take another source of light. Okay, take another source of light whose intensity is greater than the intensity which is already present. Okay, so it will be called as I plus delta I. Okay, you focus that light onto this plane. Okay, you focus that light onto this plane. You just need to keep sure that this intensity is greater than the earlier intensity, which is I. You go on increasing the intensity, we should be greater than this. For the first, delta I as 1. Okay, you observe, is there any discrimination between these two, bright light and this light? Okay, so if you are able to discriminate between these two intensities, okay, you need to discriminate between these. If there is no discrimination between these two intensities, again you increase the delta I. Okay, you do it until you are able to discriminate between this intensity and intensity. Okay, so that itself is called as Weber ratio. Okay, the amount which you are able to discriminate, okay, it's called as Weber ratio. Okay, it's called nothing but delta I divided by I. Okay, delta I by I. Okay, so if you are able to discriminate between this light and this light when the delta is I is very small, okay, when the delta I is very small, then your vision is very good. Okay, your vision of discriminating between the two lights is very good. If delta I is larger, okay, if it is very larger, okay, if it is very large, it indicates that you are not able to discriminate properly. Okay, you are not able to discriminate properly. Okay, this indicates there is no proper vision of discrimination. Okay, clear with this? Hello? Hello?
Yes, sir.
So this ratio is shown using a graph. Okay, so this is your 0, so this is your dark region and this is your bright region. Okay, so you just look over here. So here log of delta IC by I. Okay, at this region delta IC by I is what? Very high. Okay, if it is very high means what does it indicate? You are not able to discriminate properly. Okay, so this is very high over here. Okay, so this is in which region? This is in dark region. Okay, in the dark region you will not be able to discriminate properly. Okay, when you come for here, okay, so it's 0. Okay, entering into the light region. Okay, and here this is your light region. So in the light region, what is happening to here? The value of delta IC by I is decreasing. Okay, if it decreases means what does it indicate? You are able to properly discriminate the two intensities. Okay, so this is the Weber ratio curve. This is very important from point of exam. Okay, so dark iddaga you will not be able to discriminate value of brighter, you will be able to discriminate. The main thing is as delta IC by I decreases, okay, when the value is very low you will be able to clearly discriminate. When the value is very high you will not be able to discriminate. Okay, so this is regarding the Weber ratio function. Okay, alright, the next one, explain with example. Explain with example. Explain with example. Explain with example that the perceived brightness is not a simple function of intensity. Is not a simple function of intensity. So when you look at an image, okay, the image is not just a function of intensity. Okay, it depends upon various other factors. Let us it one by one. Okay, so the first thing is called as match band effect. Okay, so if you look over here, okay, this rectangle, okay, this rectangle is having uniform intensity throughout. Okay, this rectangle is having uniform intensity throughout. Similarly, this rectangle is also having uniform intensity throughout. And this is the actual intensity. You can look over here, from here to here there is actually equal intensity, from here to here there is equal intensity. But when you look at this image, what happens is there is a line, you are looking at a line which is being formed over here. Okay, this line does not actually exist. Okay, this is called as match band effect. You are able to see this line because of your visual system. Okay, this is caused just because of overshoot and undershoot of the visual system. You should have been viewing this graph instead of that. So here undershoot takes place and here the overshoot takes place. due to this overshoot and undershoot, you are able to see these lines, but actually there does not exist any line. You are able to see these lines just because of the overshoot and the undershoot. This indicates that so whatever the image you see does not only depends upon the intensity of the light, but it also depends upon the human visual system. So this effect is called as Mach band effect. Okay, it's called as Mach band effect. Similar to this, so if you look over this image, okay, the image, okay, whatever you are seeing, its intensity also depends upon the background of that image. If you look over here, all the center rectangles, okay, all the center rectangles are of equal intensity, okay, are of equal intensity. But here this is looking lighter, this is more, and this is looking darker. Okay, although all the intensities are of same intensity, okay, all the intensity of the center square is of equal intensity, you are looking it as different intensity just because of the background. Okay, here the background is very dark, so it is looking lighter. Okay, it is not so, background is lighter, so this is looking as darker. So here the image intensity is not only the function, it also depends upon the background of that intensity. Okay, so these are the two things which we need to consider. So it shows that perceived brightness, whatever the brightness which you are looking at the image, does not depends only on the intensity, but it also depends on various other factors like Mach band effect, and it also depends upon the background boundary, okay, of that image. Next, write a note on optical illusion. Write a note on optical illusion. Can anyone tell me what is meant by optical illusion? Hello? What do you mean by optical illusion? Just give me the general answers. What do you mean by optical illusion? Hello? Am I audible? What do you mean by optical illusion?
Yes, sir.
Okay, what do you mean by optical illusion? Any answers? You have viewed images, right? Optical
So,
illusion.
like it is a, so it is what?
Is it? Okay, if your eye fills in non-existing information, okay, if your eye fills in non-existing information, it is called
It
as
is a visual perception.
like a visual perception. Okay, if your eye fills in non-existing information or wrongly perceives the geometrical properties, okay, it's called as optical illusion. Okay, the main purpose for this is blind spot. Okay, the blind spot which is present in the human eye is the major reason why you'll be able to come up with optical illusions. Okay, let us look at the various optical illusions. Okay, so if we look over here, okay, so there are four circles. Okay, there are four circles. So out of all the four circles, okay, 3/4 of the circle is filled with dark, okay, or black. Part of that circle is left as it is. Okay, all the circles are done as it is. Okay, but there is no square. Okay, you are not formed the square, but when you look at this image, there is a illusion that you are written a square. Okay, there is a illusion that you are written a square over here. Okay, this is called as what? Optical illusion. So there is non-existing information, so the square is the non-existing information, so this is wrongly perceived by your eyes, so this is called as optical illusion. Similar to this, if you consider here, there are only the lines. Okay, lines are arranged in some format, but when you look at this, you see there is a circle at the center, but actually there is no circle at the center. So this is the wrongly perceived geometrical property. Okay, again if you look at these two lines, okay, you observe that this line is, the length of this line is smaller when compared to the length of this line, but actually what happens is the length of both the lines are same due to the arrowhead direction. The arrowhead is pointing outwards and the arrowhead is pointing inwards over here. Due to this geometrical shape, you are able to view that this line length is lesser when compared to the length of this line, but actually the length of both the lines are same. So this is an other example of optical illusion. Again, all the lines, okay, all these lines, center lines, are parallel to one another, but due to hashing, okay, cross hashing of this, it looks as if all the lines are not parallel to one another. Okay, but actually all the lines are parallel to one another. Okay, so if your eye fills in non-existing information or wrongly perceives geometrical properties of an object, it's called as an optical illusion. Clear with this? Hello? Hello? Hello?
Yes, sir.
Yes, sir.
Sir. Yes, sir.
Okay, read on next question.
Yes, sir.
Explain the principle of image acquisition. Explain the principle of image acquisition using explain the principle of image acquisition using a single sensor with a neat diagram. Using a single sensor with a neat diagram. So a sensor is used to capture an image. Okay, so this is a single element of a sensor. Okay, so the innermost is the sensing element, so the outermost is the box within which the sensor will be placed. So I'll be calling that as housing. Okay, energy is nothing but the reflected light which is coming out of the object. Okay, and there is a filter which is kept. So the sensor is powered by some power source which is power in. Okay, so the reflected energy, when it falls on the sensor, with the help of the power, the sensor converts this energy into intensities. Okay, into various intensities which is continuous in nature. These intensities are proportional to the light intensities, that is the reflected energy. So this is the a function of a sensor. So we may use filters. For example, if you make use of green filter, the intensity for the green light would be very high when compared to other lights. Similarly, you can make use of red and blue. Okay, whatever the filter which you use, the intensity for that particular light will be very high. This is one element of the sensor. Okay, this entire will be called as a single sensor. The main object is what? The energy has to be converted into electrical voltage waveform. So this is continuous in nature. Then we need to digitize this voltage intensity. That forms your digital image. Okay, if there is only one element, it's called as single sensor element. Elements are arranged in a linear fashion. Okay, it's called as line sensor. If it is arranged in a array, it would be called as an array sensor. Okay, clear with this? Hello? Hello?
Yes, sir.
Okay, let us move.
Yes, sir.
Okay, now let us consider, earlier you are using only one sensor. You will be using this single sensor to take the 2D image. Okay, 2D image means what? Is a function of two coordinates f of x and y. Okay, one should be, if we consider image, okay, if we consider 2D image, this is how it would look like. Okay, one would be your x direction and the other would be your y direction. Okay, So we need to move from x along, we need to move along both x direction and y direction to take the complete image. So you're having only one element, okay, you're having only one element, and this one element should capture the whole image. So what we need to do is we need to move this element in x direction also, and along with that we need to move this element in the y direction also. So what we have done is we have placed this single element on a screw. Okay, we have placed this element on a screw. So when the screw rotates, so this moves in this direction. Okay, we accomplished the movement of the sensor in x direction. Okay, with the help of the screw, what we are doing is we are moving in x direction. Okay, so now what we need to do, we need to think of moving this sensor in the y direction. So what you are doing is, instead of moving the sensor, we are keeping the film or negative film on a cylindrical object, and you are rotating that cylindrical object. If the cylindrical object rotates like this, it is nothing but movement in which direction? Y direction. So accomplished movement in y direction. Okay. By making use of a screw and making use of a cylindrical object, we are able to trace the entire image. That is a two-dimensional image. It is just by using what? A single sensor. But the disadvantage with this is there are two rotating objects. Okay, the rotating has to be controlled precisely. In order to control this, we require complex circuits. Okay, so this is the disadvantage of using a single sensor. Okay. Clear with this? Hello? Hello?
Yes, sir.
Okay, next let us consider
Yes, sir.
linear and circular strips. Okay, earlier you used only one element. So earlier you used only one element for imaging. Now we'll be using two objects. That is linear. Okay, earlier you were using only one single element, which was moved in x direction using a screw. Okay, you are using screw to move in x direction, and you are using cylindrical object to move in y direction. Now what you can do is you can eliminate the cylindrical movement. You can eliminate this cylindrical movement by keeping sensors. Okay, keeping sensors in a, when you arrange this numerous sensors in a line, it's called as line sensor. Okay, so this is your line sensor which is being used. Okay, so this covers one direction. Okay, this covers entirely one direction. Now we need to move only in one direction. Okay, so x direction. So your line strip would be kept here. So the entire strip covers one direction, and we just need to cover another direction. So we are moving in x direction. So the y direction is entirely covered by the linear strip. You just need to move in one direction. So what does it do is it eliminates the need of movement in one direction, which was done by using cylindrical object. So the complexity of the circuit would reduce. So this is the advantage of linear sensor. Clear with this linear sensor? Hello? Hello?
Yes, sir.
Sir.
Next is the circular sensor. In case of circular sensor, so this is the source. Okay, so we need to form 3D object. Here we will be using circular sensor strip to form the 3D object. Let us consider we need to take the 3D object of this cylinder. Okay, so here instead of light source we'll be making use of X-ray source. So this object, okay, from here to here, whatever you are looking, this is your source. This X-ray source emits the X-rays, okay, it will be emitting the X-rays. Whatever the circle around X-ray source, it's called as circular sensor strip. When the X-ray is emitted from this, it passes through the rod, okay, and it falls on this circular strip, okay. This image, that is this intensity, will be captured by the circular strip, okay. So this X-ray source will be rotated in one direction. Here it is rotating in one direction. If it comes here, it will be collected here, okay. Similarly, it makes one complete rotation, okay. So the opposite side will be collecting the intensity information, okay. So this is regarding the circular sensor strip, okay. Once the information is collected, it is stacked up, okay. So it is not a single information, the information will be stacked up, and then this information will be used to construct 3D object, okay. It will be used to construct the 3D object. So this is regarding the circular sensor strip, okay. So this is very important question, comparison between linear and circular strip. Next add-on, explain image acquisition. Explain image acquisition using a sensor array. So this is the sensor array. As you saw earlier, if there is a single element, it's called as element. If it is arranged in a linear fashion, it's called as a linear sensor. If it is arranged in a array format, it's called as array sensors, okay. It's called as array sensors, okay. So here this is the array sensors. Let us consider how the image is being formed. This is the source of energy. It can be sun or whatever may be. It is just a light source, okay. It's a illuminating source, okay. It is the energy. This is the object which has to be captured, okay. Let us consider this is the object which has to be captured. The light falls, okay, the light falls on this object, and the reflected light, okay, the reflected light will be captured by this imaging system. The reflected light will be captured by this imaging system. So this imaging system will be having the sensors, okay. So this will be having sensors, so that converts the intensity into continuous voltage waveform. This is the analog voltage waveform which is formed on the internal imaging plane, okay. Then we need to convert this analog waveform into digital form, okay. So this will be done by sampling and quantization, okay. Will be converted into digital image, okay. So the final output would be your digital image, okay. So the essential things are: require energy source, there should be an object which has to be captured, there should be an imaging system, and there should be an quantization unit, okay. If this is a camera, okay, if this is a camera, at the front end there will be a lens which will be collecting the intensity of light. At the back end there will be sensors which will be converting that intensity of light into voltage waveforms. But this would be continuous in nature, that is analog in nature, which has to be converted into digital form. This is done by sampling and quantization. So clear with this? Hello? Okay. Next, explain the process of generating digital image. Explain the process of generating a digital image, and explain the concept of sampling and quantization. Explain the concept of sampling and quantization in image processing. In image processing with an example. In image processing with an example. So let us consider this in the next class. Okay.